Hello and welcome. Today we will be learning about how to retrieve sequences, whether DNA or protein, from NCBI. First, we have to go to the web browser, Chrome or Safari, anything. Now in this section, we have to type NCBI. Now clicking on NCBI will display us the home page of NCBI. Now in this all databases section, I have to click on nucleotide if I want to retrieve the nucleotide or DNA, basically the DNA sequence. Now here in this section, I have to type the sequence name or the gene name, then press on the search or simply press enter button. Now it will display me a number of sequences. Now if I select on the first sequence, it will display the sequence. Now the sequence here are basically in two formats, GenBank and FASTA. This here is displaying the GenBank format. GenBank format is more descriptional. It has a lot of information to give. For example, here you can see that along with the sequence here, a lot of other information like the accession version, the organism from which this sequence was derived, also the reference authors, like who submitted this sequence has also been given, and the title of the journal where this sequence was primarily submitted. For example, this Agrawal VK et al. Have submitted this sequence and it was uh, submitted to Molecular Genetics Laboratory in the Animal Genetics and Breeding department in the College of Veterinary and Animal Sciences, Rajuvas. So this is the detailed information GenBank will give you about any particular sequence. Now it is also telling us the sequencing technology that has been employed, like for this particular gene sequence, Sanger dideoxy sequencing method has been used. Now it has been taken from the organism Bos indicus, and the genomic DNA was particularly used for sequencing this particular gene, and the breed from which the this blood sample was collected is Sahiwal. So this is all the information that GenBank provides us, and here is other informations are also given like gene, mRNA, and CDS, coding DNA sequence. If I click on this coding DNA sequence, it will display me the coding DNA sequence of this complete gene. Okay, this was about the GenBank format. Now, if I want to see the FASTA format, which is a little bit different from the GenBank format, I have to click on the FASTA. It will display me the FASTA format. Now, FASTA format, as you can see here, it starts from a greater than sign, then the sequence identifier. It is telling us that it has been retrieved from Bos indicus, breed is Sahiwal, and the sequence is particularly of the DGAT gene, and it is representing partial CDS. So this is the complete sequence. Now, if I want to download this sequence, there is another thing, like you can visualize this sequence in the graphics format as well. So this is the graphic format like where particularly your gene will be there in the genome of this Sahiwal breed. So here is the gene DGAT. Now it will tell you the exact position as well as the number of nucleotides. Here you can see that 411 nucleotides, and you can look for different features of this gene as well, which are the genes that are upstream, which are the genes that are downstream, so that you have the idea about the complete location of this particular gene. Now coming to retrieving of this sequence, if I want to retrieve the GenBank format, I have to go to GenBank, or if I have to retrieve FASTA format, I have to go to FASTA. Now once I get this FASTA format here, I have to go to send to option, select file, select the format. If I want GenBank, I can select on GenBank. If I want FASTA, I can select FASTA. After selecting FASTA, I have to click on this create file. This will create a file of this particular sequence which I can visualize. So here's the FASTA sequence that I've already downloaded. Now I can use this sequence for designing primers or any bioinformatic analysis. So this was about retrieving sequence from NCBI. Thank you. See you in the next lecture.